Antigen-binding B cells are trapped in the T cell zone of secondary lymphoid tissues and are activated by encounter with armed helper T cells. One of the most puzzling features of the antibody response is how an antigen-specific B cell manages to encounter a helper T cell with an appropriate antigen specificity. This question arises because the frequency of naive lymphocytes specific for any given antigen is estimated to be between 1 in 10,000 and 1 in 1 million. Thus, the chance of an encounter between a T lymphocyte and a B lymphocyte that recognize the same antigen should be between 1 in 10 to the 8th power and 1 in 10 to the 12th power. Achieving such an encounter is a far more difficult challenge than getting effector T cells activated, because, in the latter case, only one of the two cells involved has specific receptors. Moreover, T cells and B cells mostly occupy quite distinct zones in peripheral lymphoid tissue. As in naive T cell activation, the answer seems to lie in the antigen-specific trapping of migrating lymphocytes. When an antigen is introduced into an animal, it is captured and processed by professional antigen-presenting cells, especially the dendritic cells that migrate from the tissues into the T cell zones of local lymph nodes. Recirculating naive T cells pass by such cells continuously, and those rare T cells whose receptors bind peptides derived from the antigen are trapped very efficiently. This trapping clearly involves the specific antigen receptor on the T cell, although it is stabilized by the activation of adhesion molecules and chemokines. Ingenious experiments using mice transgenic for rearranged immunoglobulin genes show that in the presence of the appropriate antigen, B cells with antigen-specific receptors are also trapped in the T cell zones of lymphoid tissue by a similar mechanism. On encountering antigen, migrating antigen-binding B cells are arrested by the activation of adhesion molecules and the engagement of chemokine receptors such as CCR7, a receptor for MIP, 3-beta, and SLC. And that's antigen-binding B cells are trapped in the T cell zone of secondary lymphoid tissues and are activated by encounter with armed helper T cells. Have a great day.